So today we're going to start Unit 7, Lecture 3, where we're going to focus on carbon metabolism. So this is also referred to as methylation. This includes our B6, our folic acid, and our B12. So first we're going to talk about folate or folic acid. It's also referred to as B9. And you probably know folic acid mainly due to the encouragement of women that are pregnant to take higher levels of folic acid to prevent spinal bifida. So we'll talk about when we get to deficiencies, specifically be discussing why we have this deficiency in folic acid causes spinal bifida and other neural tube defects. So folic acid was initially discovered in its association for preventing megaloblastic anemia and neural tube defects during pregnancy, and it's found pretty highly in our green leafy vegetables, so that's why it is called folate, because it came from foliage. And it also is works in the same cycle as our B12 primarily. So a lot of the deficiencies in B12 and folic acid look very similar, so it's hard to distinguish. So here we have our structure of our folate in the various forms. So this main picture on the top, you can see what is making up our folic acid. And the main difference between our folate, our dihydrofolate, and our tetrahydrofolate all depends on the amount of hydrogens that are on this 7 carbon and 6 carbon. If we have our hydrogens, 2 hydrogens on our 7 carbon, it becomes dihydrofolate. Where if we add an additional hydrogen onto the 6 carbon, it becomes tetrahydrofolate. And tetrahydrofolate, this is our THF. This is our active form. Now, when we go to absorb our folate, we do have different forms. So we do have these polyglutamate forms. So poly meaning that it has more than one glutamate chain. So there are multiple glutamates. So when those are in the intestine, we hydrolyze those to our monoglutamate form. So this PTM is actually our folate. And this happens using our gamma carboxypeptidase. And that's coming from the pancreas. And then once we're inside, our folate is able to be absorbed in the duodenum and the jejunum, and this is via active transport. So we have a sodium dependent active transport. And this is also similar in that we do have some passive transport, and passive transport occurs just like all the other ones if you have greater than adequate amounts. So our sodium dependent will occur when we have low to adequate. So greater than adequate is going to be our passive transport. And then once we're inside the cells, we end up reattaching the glutamate residues to form our THF. And it circulates in the blood as our monoglutamate derivative. So we only have one glutamate. In the pigs, it can circulate as THF, and most circulation is free, but we do have some non-specific binding to albumin, and we do have some folate binding proteins. Our folic acid within the cells are polyglutamine forms, so that prevents any passage out of this membrane and it catalyzes the enzymatic reactions more efficiently, having that additional glutamate on the end of it. So our THF is a cofactor coenzyme for enzymes that are involved in one carbon transfers, and this is our methyl group. So we are changing one methyl, transferring one methyl groups to different compounds. For this, we have to have B12. So folate and B12 work together. And B12, as we'll talk about in a little bit, is a coenzyme for the methylation cycle referred to as SAM. So here we have a summary of folate's function in this one carbon metabolism. So you can see there's actually 3 cycles. We have our SAM cycle down here. We have our folate cycle up here. And then we have our purine or pyrimidine cycle here. And you can see that we need folate. It is remethylating B12. And we need this for the resynthesis of our methionine and homocysteine. And when we have this methionine going back to homocysteine, we are actually doing this so we can have DNA methylation. And so this is post-translational. And then we have over here with our purine and pyrimidine synthesis, you could see also here that we have our folate is needed for the synthesis of our pyrimidines and purines, and this is for actually making our DNA. And the areas in which these enzymes are darkly circled, so our methionine synthase, our RCF, so this is a transporter of folic acid into the cell, and then we have our MTHFD and our MTHFR. So this is reductase, dihydrogenase, and our thymidate synthase. All of these have inborn genetic errors. So we have places in which genetic mutations that are resulting in these enzymes being less efficacious. And primarily in the case of the neural tube defects, that is primarily using the MTHFR, to where about 5% of all women have a mutation within this MTHFR. So basically, this requires a higher amount of folate in the diet so we can overcome the low affinity of the MTHFR. So if we don't have this working adequately, we're going to not be able to regenerate our folate, and therefore we end up dramatically affecting our DNA methylation, our post-translational DNA methylation, and that's really what's causing the neural tube defects. So the main deficiencies in humans, you're going to see it primarily clinically in megaloblastic anemia. So this is where individuals, if you look at their red blood cells, you do a blood smear, you're going to see the red blood cells are much larger than you anticipate. In addition, the white blood cells are going to be larger and going to have more DNA. And that occurs because these red blood cells are remaining in an immature state. And if you know anything about red blood cells, especially in humans, they typically do not have a nucleus. So these red blood cells actually end up having some level of nucleus remaining because they are immature and haven't had the nuclei broken up yet. So here we have a picture of our megaloblastic anemia. You can see that the red blood cells are unusually large. They look more more roundish, not as much disc form. So that's one indication that you have a folate deficiency. So here we have a baby with anencephaly. This is due to the lack of the formation of the frontal lobe, and it occurs early on in gestation. So basically, we don't have the proper closure of the cerebellum. So as I said, the majority of these babies are stillborn, or they only survive for a few minutes after they're born. Some doctors, when at the 20-week appointment where you have your ultrasound, they can see that these babies do not have the development of their brain, and they may actually encourage an abortion because the baby will not survive. And our poultry are actually the most susceptible of all the domestic animals for folate deficiency. You will observe poor feathering, poor growth, cervical paralysis, porosis, so that's that slim tendon, and anemia in the poultry. So here you can see the two on the left. So we have this chicken and this chicken. They both have folate deficiency, and you can see the paralysis, so they are unwilling to stand up. In addition, they have poor feathering here. So that's a very good indication that we have folate deficiency in the flock. So here's another chicken with paralysis. You could also see the poor feathering that it has. It just does not look very happy or healthy. And here we have another one. You can see on the left-hand side, the folate deficient. We have stunted growth, poor feathering, compared to the control bird. So these are both the same age. So you can see how stunted the growth was and the feather was. So our folate has really not been shown to be toxic in most animals. It has a very low LD50, so it takes about 500 milligrams per kilogram to get a LD50 in rats. So that's pretty significant. So in all intensive purposes, we could say this is not toxic. The main problem with taking a substantial amount of folic acid is that it may actually mask a B12 deficiency. So it delays the onset of anemia, but you still can have the neural tube and other neurological symptoms that occurs. So next we're going to move on to vitamin B12. So B12 is the largest vitamin out of all of them. It was also the last to be described, so it wasn't described until 1948 when we determined the configuration. And one of the main things, it contains cobalt, the metal ion cobalt. The names, if you ever look, it may be referred to as cyanocobalamin and methylcobalamin. So the cobalamin is the indicating that it has cobalt in the midst of it. I would like to state that ruminants do not have a B12 requirement. They require cobalt because the microbes are able to synthesize B12, but they need to have the cobalt within their diet in order to synthesize it. So you can see here how complex the structure of B12 is. The main thing here, we have cobalt in the very center. With this, you're definitely going to have to tell me that cobalt is the center mineral that is making up the structure of B12. Also, I want you to be able to tell me that it is the most complex and largest vitamin that is needed.
So out of all of our B vitamins, B12 is by far the most complicated and tricky for digestion and absorption. So where all of our other B vitamins are microflora can synthesize them, that is also true for our B12. However, our B12 needs these extrinsic and intrinsic factors in order to be absorbed. So when our B12 first comes through our food, it is actually bound to an extrinsic factor. It's found in our foods. So not an intrinsic, but an extrinsic, so a non-intrinsic factor. And once our B12 goes into our stomach, it's released from that extrinsic factor, and that is done by our hydrochloric acid. So another function of our hydrochloric acid that we are observing. And then we need that B12 to bind what we refer to as intrinsic factor. And this is the whole reason why we have to have B12, so why a lot of hindgut fermenters are actually practice coprophagy, is for them to obtain their B12. And that's because it has to be bound to the intrinsic factor in order for it to be absorbed. So this is, it binds to intrinsic factor in the stomach, and there is an intrinsic factor receptor that's located in the lumen of the small intestine in the ileum. And we have 70 to 80% of our B12. So once it's absorbed, conjugated to that intrinsic factor, 70 to 90% of it is transported as adensylcobalamin and methylcobalamin, and it binds the transporters transcobalamin 1 or transcobalamin 2. Now, the transcobalamin 2 is actually better because it has a higher turnover rate, making B12 more readily available for the cells, where the lower turnover rate of transcobalamin 1 means it's going to hold on to the cobalamin. Greater has a higher affinity, so it's not going to release and give it up to the cells as easily. So here we have our dietary protein, which is our red triangle here that comes in through our food, and it enters into the stomach, and there are actually two binders of B12. So we have these R binders and intrinsic factor. If you notice, intrinsic factor is actually being released from the parietal cells. So this is somewhat competing for the binding of B12. So then as soon as our B12 has a higher affinity for these R binders, and it then goes into the small intestine, and it's here that we actually utilize trypsin, which is going to break our B12 off of our R binder, and then that's allowing it to bind intrinsic factor. So these proteases, this is actually trypsin, is the main one that is affecting it. So now we have within our duodenum that our B12 is binding our intrinsic factor, and then once that is bound together, we have within the distal ileum, this is binding the intrinsic factor, and then we have it absorbed, and you can see the primary one is transcobalamin 2. This has a lower affinity, so it's more willing to give it up, and we have about 50% of that is taken to the liver, and an additional 50% is taken to the other tissues. So as you probably gathered from the folate portion of this lecture, that B12 and folate interact significantly. That's one reason why we have so many of similar deficiency symptoms, and it's hard to distinguish between a folate deficiency and a B12 deficiency. So some of the things that B12 is responsible for is it is accepting a methyl group to activate the B12 in its coenzyme form, and that is allowing the methylation of homocysteine to methionine. So that's where we have, this is part of our SAM cycle. So we're regenerating our methionine. In addition, B12, because folate is responsible for having our synthesis of our purine and pyrimidines, and we need B12 to regenerate our folic acid, or THF. B12 also ends up participating in the regulation of DNA expression, mainly because of the purine and pyrimidine synthesis that it's involved in, that we talked about during folic acid. And it also transfers those methyl groups, and that is maintaining the integrity of the myelin sheath within the nervous system. So if you have someone with B12 deficiency, they may have symptoms more similar to Parkinson's disease, which is associated with an inadequate amount of the myelin sheath. So here we have another version of that interaction of all those different cycles, but we're primarily focusing on the folate cycle and the SAM cycle, and because this is the main place in which B12 is involved. So our B12 is specifically required for our methionine synthase. So we utilize this to regenerate our methionine. So we have our homocysteine combines with our 5-methyl tetrahydrofolate. So our B12 is donating a methyl group to the methionine synthase, and we're able to generate our methionine, and we also get the resynthesis of our THF. So we get regeneration of methionine, regeneration of our THF. So basically, if we don't have our B12, we are not going to regenerate our THF, and therefore we're going to have over here the other component of the cycle is going to be our purines and pyrimidines, and so we're not going to be able to synthesize that, which is going to affect our DNA synthesis. So because of that, both our B12 and our folic acid are going to result in megaloblastic anemia, and resulting in a significant amount of DNA immature state. So the cells start to undergo mitosis, but they're not able to because there's inadequate purines and pyrimidines. So then they're just stuck in this level, but your body needs those red blood cells to transfer the oxygen, so you result in just having immature red blood cells circulating around. So what are the main causes of B12 deficiency? Primarily, it's going to be the lack of red meat or really any animal protein. So we do have us humans, we are omnivorous, so this is really an indication that you need to have some sort of animal product so you can actually meet your B12 requirement. Now, B12 is actually the vitamin that you need in the lowest amount, so it really does not take very much at all to meet your requirement. So if you are vegan, you need to ensure that you are taking a B12 supplement. Otherwise, you will result in a deficiency. If you have atropic gastroenteritis, so this is where you have a lack of hydrochloric acid and intrinsic factor, you have atrophy due to the gastric glands. So this really happens quite commonly in elderly individuals. So the older you get, the more likely that you will need to get a shot of B12. Um, as with all elderly people, they become less and less efficient at absorbing many of these things. One of the other reasons why elderly people really, when they get up to 80 plus years, you really see them start decreasing the amount that they're consuming, and that's because of this atropic enteritis. So they just have really slow passage rate and get full really easy. And so we have a lack of hydrochloric acid, a lack of intrinsic factor. So then if we don't have intrinsic factor, we're not able to bind the B12. We don't have the hydrochloric acid to break the B12 that is there from the extrinsic factor. So therefore, we end up getting anemia. So you have perinacea anemia that results due to the atropic gastroenteritis. You get peristalsis of the nerves, tremors, lack of coordination, and a lot of that has to do with the effects of the lack of B12 on the myelin sheath. In poultry, you see a reduced feed intake and weight gain, nervous disorders, defective feathering, and leg weakness and porosis. So many similar things that we saw with our folic acid deficiency. So here we have a chicken that has both poor plumage and significant amounts of leg disorders, so they're unable to walk. And the leg disorders is occurring more so due to the effect on the myelin sheath than it is to do with porosis or slip tendon. Next, we're going to talk about vitamin B6. So our vitamin B6, in addition to our vitamin B12, these are the only two of the B complex vitamins that actually go by their old numbering system. And we have, and it's mainly because we have multiple types of B6. So we have 3 naturally occurring forms, and all of these are interconvertible, and there are a total of 6 compounds altogether that have B6 activity, but we're only going to talk about the naturally occurring forms. So the first one is pyridoxal, which is abbreviated PAL. So knowing your organic chemistry nomenclature, the all is aldehyde form. And this is the form that's going to be primarily found within your vitamin supplements. And then we have pyridoxine, which is abbreviated as PN, and pyridoxamine. It is a coenzyme for pyridoxal phosphate and pyridoxamine phosphate, so PLP and PMP. And it is critical for several areas of metabolism, anything that needs transamination or decarboxylation. It's also involved in glycogen phosphorylation, and it's important for steroid hormone action. And we can find it in a variety of food sources. So it's found in meat, vegetables, nuts, cereal, grains. It's primarily in our cereal grains found within the germ and the alaneric layer. And basically our B6 gets removed when we are refining those or milling those cereal grains, but there are adequate amounts within the other ingredients in the diet. So the structure of B6 with these different chemicals, it really depends on the source. So the primary source from plants is going to be the pyridoxine, and this is also the most stable out of all the B6 compounds. And then our animal products are going to have pyridoxal and pyridoxalamine. And you can see here the differences. So Paradoxol has this aldehyde. We have the hydroxyl group on our paradoxine, and then we have our amine group on our paradoxamine. The critical thing for all three of these structures to be active is we have to be able to phosphorylate at the 5-hydroxyl methyl group. So here we need to be able to add a phosphate group onto there, and then we're able to call it a fully functional B6. So definitely know that at the 5-hydroxyl methyl group, we need to add a phosphate molecule onto it to make it active. So when we consume our diets, the majority of our B6 is actually going to be bound to protein in its phosphorylated form. So it's necessary for us to dephosphorylate that before the B6 can actually be absorbed. And all three forms can be absorbed by passive diffusion. And then we have paradoxine and paradoxal are phosphorylated in the cytosol. This is dependent on paradoxal kinase, so it's AT dependent paradoxal kinase, so it is requiring some energy. And then it is also absorbed in the jejunum and the ileum. And once it's absorbed, we have 90% of it is transported in the blood, and it's primarily bound to albumin or on the erythrocytes, which is also known as our red blood cells. And it actually binds albumin. So once we have our B6 cross into the cell membrane of the cell that is taking it up, so here we have paradoxal, we quickly convert it to the phosphorylated form, and we're utilizing PAL kinase. And if you remember, our kinases are always adding a phosphate group on. And we're able to store small quantities in the body, so where some of the other B vitamins we're not really able to store, we can store small quantities. And the majority of that is primarily contained within our muscle. So our muscle is storing about 70 to 80% of our B6. Any excess B6 we're going to excrete in the urine, and it's primarily excreted as 4-paradoxic acid. And then we have various compounds that are going to increase the loss of B6. So amphetamines, risperidone, alcohol, oral contraceptives. So if you happen to be taking any of these particular drugs, you actually may want to make sure that you're taking a multivitamin, because you will be excreting more of our B6 and have a slightly higher requirement because of that. So our B6 has a lot more functions, so it's significantly involved in our 1-carbon metabolism, but unlike folic acid and B12, it has significantly more functions than just that. So it's actually a coenzyme with many of our nitrogen-containing substrates. So it's pretty significantly involved in our amino acid metabolism. We need it for over 140 reactions, so anything where we have to transaminase, decarboxylation reactions, if we have beta group interconversions, all of that requires a B6 coenzyme. And it primarily is forming a Schiff base when it binds the enzyme. So some of the functions that B6 is necessary for, and I've mentioned this first one previously when we were talking about niacin. So we need B6 to convert tryptophan to niacin. So if we have inadequate B6, we're going to, and we're relying on our tryptophan to meet our niacin requirement, we're going to end up getting some form of niacin deficiency, most likely in the form of pellagra. Because it's involved in all these transamination reactions, it's necessary for gluconeogenesis. We need it to utilize and free up our glycogen, to break down our glycogen to increase our glucose levels. It's also necessary for neurotransmitter synthesis. So we need it to make serotonin, we need it for epinephrine, and we also need it for norepinephrine. When we're making histamine, so you can actually think if you have allergies, especially during the heavy pollen times that you encounter in spring, sometimes in the fall. So our B6 is necessary to make adequate levels of histamine, and that's what's actually giving you those lovely springtime allergies. And then it's also significantly involved in our DNA methylation. So if you have inadequate B6, because it's involved in these folate, SAM, and pyrimidine reactions, it can actually cause mutations because it's involved in our post-translational DNA methylation. So here we just have a few of the reactions that it's involved in with our neurotransmitter synthesis. So converting our glutamate to GABA, we need our B6. When we're converting tryptophan to serotonin, we need it to convert our 5-hydroxyltryptophan to serotonin here in the second reaction. In addition, utilizing our tyrosine to make our dopamine, which is then converted to norepinephrine and epinephrine. We also require our B6. So our vitamin B6, it is involved in transferring the methyl groups around, just like folic acid and our B12. The one thing that's missing from this picture is it did not mention the B6 that's required from going from THF to the 5,10 methylene THF. So it transfers methyl groups to folate, so our THF. We now add a methyl group, so that's how we get the 5,10 methylene THF. And then folate then goes and transfers those methyl groups to our B12. And then those methyl groups are transferred on to our methionine. So it's almost a nice little figure eight that's occurring. So a B6 is converting to folate, and then our folate is then taking that and generating our methionine. Our methionine is making homocysteine, and then we get more THF. So it's this big figure eight. In addition, other places that our B6 is involved is just generating our cysteine and alpha-ketoglutarate. So when we say that we can utilize, as we mentioned earlier in the semester, that we could utilize methionine to synthesize cysteine, well, that's going through the SAM cycle. So if we have excess levels of methionine, we then will make homocysteine. Any homocysteine that we make in excess, we're able to take out of this cycle, and we convert it and we make it into cysteine and alpha-ketoglutarate. And then that could go on to make our glutathione. But here we have our cysteine. So without our B6 is required for these two reactions to synthesize cysteine from our homocysteine. So without adequate B6, we're also going to limit our cysteine synthesis if we're utilizing methionine as the primary source of our cysteine. So with our B6 deficiency, I'd like to mention first that it's pretty rare in humans. Usually if you're going to see B6 deficiency, it's primarily caused by alcoholism. And with those individuals, you get some sort of rash or neurological change. Since B6 is required to make those neurotransmitters, that's why we're having neurological issues. We got hyperkeratotic scaling, dermatitis, our weakness, our muscles are very weak, you're irritable, difficult time sleeping, and in severe cases, because you have the lack of the neurotransmitters, we can induce seizures with a B6 deficiency. So here we have some chickens that we induced B6 deficiency in. So you could see off of this top picture that our chickens have very slow growth. They have very poor feed intake. You'll encounter them running aimlessly with a drooped head, and a lot of that has to do with the lack of neurotransmitters. Porosis, so you'll get that slipped tendon, you'll get edema in the eye, rough plumage, and paralysis. Pigs, you'll also see reduced growth. So here we have a picture of two pigs, the one on the right over here. So this one is actually deficient in B6. So they're the same age, and you can see how much smaller they are. So they have reduced growth, and they also have alopecia, which is the loss of hair. So this one, it's hard to tell in this picture, but it has less hair on it than the other one that is adequate levels of B6. So B6 toxicity isn't really too common, but it can be toxic at high levels. So toxicity was first reported back in the early 80s, and that's because women were taking large amounts of that to treat PMS symptoms. And it wasn't until around that time that PMS was actually classified as a true medical condition, and they've found that higher doses of B6 actually reduce the severity of a woman's PMS. But with these high levels, and this is greater than 2 grams per day, so this is a significant amount of B6 that they were taking. They had some neuropathy, and these symptoms ended up reversing once the women stopped taking the supplement. You have to take a lot of B6 for it to be toxic. So the reality is you naturally aren't going to be taking that much. So this is really only seen when you have a supplement for treating PMS primarily. So some of our animal models, rats, rabbits, dogs, they tolerated a significant amount without any ill effects. But I also don't know what form of B6 that they utilized in those studies. We do know that paradoxal is twice as toxic as paradoxine or paradoxamine. So you have to be aware and cognizant over which type you are finding in your supplement. So if you're willing to try B6 as a treatment for PMS, make sure you take paradoxine or paradoxamine as opposed to paradoxal. So that's basically all we have on our 1-carbon metabolism. 3 main vitamins, B6, B12, and folic acid. So I hope that with this you can, at the end of this lecture, I want you to be able to explain how all these interact and why you end up getting those particular symptoms that you get, predominantly how a deficiency in B12 will cause deficiency symptoms of folic acid, and how B6 interacts with all of this.